Well, welcome everybody. It's a great pleasure to be here tonight. I'm Marc Carrier. I'm the president of Thrombosis Canada, a hematologist at the Ottawa Hospital. I do venous thrombosis for a living, and today my co-chair is Ryan Kerzner. Ryan is a pharmacist that got his BSc at the University of McGill in 1999, and then his pharmacy degree in 2007 and an MSc at the University of Montreal in 2008. He's been doing the anticoagulation stewardship at the Jewish General Hospital. He has a lot of knowledge on anticoagulation in general, and given his pharmacy background, he's much smarter than most of us on biosimilars. So it's a great pleasure to have him with us tonight. And we'll be discussing biosimilars is imitation, the sincerest form of clinical efficacy. And there was a scientific planning committee, so Dr. Peter Gross from Toronto was part of it, Dr. Lena Castellucci from the University of Ottawa, Ryan, our co-chair from McGill University Jewish General Hospital, and Dr. Otto Moodley from Saskatoon as well. The presenters today are myself and Ryan, and these are our disclosures. This program has received financial support from Value Pharma in the form of unrestricted educational grant. This program has not received any in-kind support. The potential conflicts for tonight are the presenters are eligible for honoraria from Thrombosis Canada, and Value may benefit from the sales of a product that may be discussed in this program. However, keep in mind that the content of this program was developed by the Planning and Steering Committee and peer-reviewed by Thrombosis Canada members. The recommendations involving clinical medicine are based on evidence that is accepted within the profession, and all scientific research referred to, reported, or used in the program in support or justification of patient care recommendations or conform to the generally accepted standards. So the program learning objective for the next 45 minutes or so is to describe at a very high level how evidence for biosimilar is assessed by regulatory agencies in Canada, discuss criteria in evaluating choosing biosimilar low-molecular heparin agents, and provide effective education on biosimilars to patients, because that's an important component of a discussion with our patients. With that, I'll pass the command to my co-chair. Go ahead, Ryan.
Thank you so much. Good evening, everyone. Thanks for joining us tonight. So we'll dive right into it, and we'll kind of, I'm going to start off with talking a bit about an overview of biosimilars in terms of what they are, what it means, and then Dr. Carrier after we'll sort of approach the thrombosis side of biosimilars as we get through the day. So the evening. So I will just, oops, there we go. So we're going to start with the test your knowledge question. So there could be more than one answer for this question, just to let you know, and we're going to open up the poll shortly. So which of the following characteristics apply to biosimilar drugs? So A, they're large complex molecules. B, contain the identical medicinal ingredients as the original brand drugs. C, made from living organisms or from their cells rather than chemicals. Or D, manufacturing involves intricate biotechnological processes. So the right answer is we actually have three of them that are correct. So these are, and we'll go over this over the next few slides, of course. So basically, the bottom line here is biosimilars are not identical to biologics, but they must be shown to be highly similar without any clinically meaningful differences in terms of safety, efficacy between them to be approved by Health Canada. Now, biologic medicines have really revolutionized the treatment of many diseases. And the first one that probably everyone knows about is insulin. So insulin is considered a biologic drug here. It's been around for a long time. And of course, it's revolutionized the treatment of diabetes. Now, a biosimilar is a biologic drug that's proven to be highly similar to a branded biologic drug. And they have to be manufactured in the same regulatory standards, so to maintain safety, efficacy, and quality as the branded biologic drugs. So we'll start with some definitions. So when we're talking about biologic drugs, we're really talking about large, complex molecules. They're made from living organisms or from their cells. And because of the way they're made, there's this natural variability. So It's, when you have, for example, if we're talking about enoxaparin as an example, as a biologic drug, it's not like a single, it's, there's many different strands of many different sizes. It's not like a single product, let's say like giving apixaban, which is a single molecule. So there's a certain variability just because of how they're made. And when we're talking about the original biologic drugs, here we're talking about either the reference drug or the originator. So on the right, you can kind of see some examples, and there's many drugs used, for instance, in rheumatology and in GI, in cancer. And so you see some examples here on the right side. When we're talking about biosimilars, we're talking about a version of a branded biologic drug, which is similar in terms of quality, safety, and efficacy to the originator, but it's not identical. And here, for example, these are the first 3 biosimilars to enoxaparin. There's a 4th one, which Dr. Carrier will allude to a bit afterwards as well. At the time we did this, there were just 3 initially. And so there's many different, so as you'll see with, there's more and more biosimilars available to this day right now. And there's another concept called bio-better, which is actually when you take your original biologic drug and you change around the molecule or you change around the process, that it actually becomes better. And these almost always precede the biosimilars. And because you make some additions or you made some modifications, they can be subject to patent protection. And there's a couple of examples here. For instance, trastuzumab and rituximab both have bio-betters that are on the market here in Canada. So that's the overview of a biologic drug. And then we have our classic chemical drugs. So these are small molecule drugs created synthetically. They're simple molecular structure that's easy to copy. So example, the original aspirin by Bayer was a chemical drug. And of course, now there's many, many different generics, of course, available. There's probably dozens and dozens of acetyl salicylic acid. So it's the generics. So everyone kind of knows what the generics are. So these are basically identical. They have identical medicinal ingredients to the reference drug. There could be some differences in the excipients of the pills, but the actual medication itself is the exact same. So biologics, chemical drugs. Now it's important to mention the biosimilars are not generics. They're really 2 different things, and people kind of confuse the 2 because it's, you think, oh, it's another company coming in to bring a copy of the drug, but they're not actually the same thing. So generic drugs are small molecules, as I said, they can be synthesized chemically quite easily. They contain identical medicinal ingredients as the reference product. But biosimilars, there's no way for them to be exactly identical to the original drug just because of the process in which they're made. Biosimilars, as I've said, are so they're basically complex, made from living cells, not from chemicals. There's a natural variability. They're produced by complex manufacturing processes. But when you're talking, another key difference is that when you're bringing them to market, there's a lot more clinical comparative studies that were required for Health Canada to approve them, or other regulatory agencies as well. So basically, you just have to show that this biosimilar that you're trying to bring to market is highly biosimilar to its reference drug. And we'll talk about sort of what that means soon. So why are we talking about biosimilars? Well, let's open up another poll question. So biosimilar drugs are used in which of the following? Vaccines, blood and blood components, allergenics, gene therapies, recombinant therapeutic proteins, or all of the above? So 83% of you got the right answer. It's all of the above. And so it's really, we kind of see these in a wide range of products. And so biologic medicines have really altered the way in which severe chronic and disabling diseases are treated. They're highly specific, developed as targeted medicine. So for instance, now in oncology, that's where you really see the biologics exploding. So you see it in different fields, in rheumatology, in dermatology, Crohn's and colitis, with GI diseases, immunology, diabetes, as I mentioned as well. So it's really a whole range of products where we see these types of drugs. So they treat many, many rare conditions and also involved in the prevention of severe disease in really multiple therapeutic areas. Now, if we look at the data, so this is some data from the previous decades, from 2011 to 2020, we could see that from 2011, where we had like $3.3 billion of sales in Canada of biologic drugs, that tripled to $10 billion by 2020. And that's continued to go, that continues to rise year to year, especially, more and more, and as more and more of these become available. So if we look in 2011, this was about 17% of our drug expenditures in this country. And by 2020, that was already 34%. So these drugs do cost a lot of money, and we're using more and more of them. Now, the availability of biosimilars has been going up as well. So if you look at this graph here, we could see, and sort of Europe always took a bit of the lead on us. So in dark blue, we have the EMA, which is the European Medicines Agency. We have the FDA for the US in orange, and Health Canada in red. And we could see that so we only had one biologic drug. This is the number of biologic drugs that actually had at least one biosimilar, and there could be more than one biosimilar available. And that really started to skyrocket, especially in the final few years of that decade. Now, and I could tell you, actually, the last time I looked, we have in Canada at this date, we're close to 60 different biosimilars that are on the market in Canada right now, for about a total of about 20 biologics, 20 to the low 20s number of biologics. Now, the presence of biosimilars hasn't really changed our spending too much just yet, although that is something that's rising. There was a bit of a slow uptake. So it didn't have a huge impact, but as more and more biologics lose their patent, we're expected to have more and more biosimilars on the market, which we're hoping would have a dent in our total expenditures for healthcare. So what's the rationale? Well, really, in the end, for biosimilars, we want to try and decrease healthcare costs and increase the access to healthcare. We want to, we all know that all of our provincial jurisdictions are all strapped for cash when it comes to healthcare spending, and everyone's looking for ways to save money. Well, when we look at how much we spend on all these biologic drugs, you can kind of see why there's a need to have more and more biosimilars so that we can actually treat more patients with, and offer these treatments to patients. And in the end, it comes down to if the payers are willing to pay for this switch to biosimilars, and then from on the provincial level, on the private level, and then we'll be able to really help to maintain the sustainability of our healthcare system. So in the end, yeah, I kind of already said all this, so I'll just go on to the next one. So if we look globally, and we look at the types of cost savings, so you see in Canada here, so for instance, this was just in 2018, and only for 4 different biologics. So with filgrastim, infliximab, etanercept, and insulin glargine, there was a modest savings of modest in the overall budget of $94 million Canadian in that year alone with just those 4 drugs. And it depends how you look at the numbers. So if you look in the US, so in 2019 alone, there was a savings by switching to biosimilars, there was a saving about $2.2 billion US. And then you can see sort of other savings around in Europe, in Japan, and South Africa. So this is something that's really sitting on a global level. It's a way to be able to offer these drugs at a lower cost. So in Canada, and from the last time this presentation was given, there's been some updates. So if we look, there's pretty much almost all provinces right now, with the exception of Manitoba and the territory of Nunavut, where basically if there is a biosimilar, that's the one that's going to be covered by the provincial health system. Now, there might be different ways in which they do that, but generally, if it's, depending on the patient population, that's the one that needs to be covered if it's available. And we're seeing some changes also in the private payers as well. So each kind of insurance company sort of decides on its own if they're going to cover, and now more and more the insurance companies are really going in that direction. So if patients do prefer the original, they would have to pay that difference in general if they want access. So in the end, now my job is to show you if these are actually similar enough to each other. So let's compare a biologic and its biosimilars. So biologic medicines, as I mentioned earlier, are produced in living organisms using really cutting-edge technology. The process in which they're made are they're much more complex than producing our regular chemical drugs that are obviously bigger in number. But due to the complexity of biologics and their biosimilars, they cannot be created or produced by conventional methods. So it's really specialized technology to make these kinds of drugs. And so it's important to sort of have a bit of an understanding of what the challenges are in producing these drugs, and that'll help us understand a bit sort of how they could be shown to be similar, the biosimilars to the biologic medicines. So by using living cells, there's a certain natural variability that comes. So you get this sort of micro heterogeneity that's present, even within a batch, but also between batches of different biologic drugs. So as an example, if you produce a syringe of enoxaparin, and you have another syringe of enoxaparin side by side, it's not exactly identical from what's in one syringe to the other, but you hope the overall effect of that drug is the same. So that you have the same anti-10A effect as an example. Now, there's a certain degree of variability that's permitted, but it has to fall within a range that's outlined by Health Canada when these drugs are approved. And there's very strict manufacturing controls required to ensure that you have this batch-to-batch consistency between them. So during a life cycle, there's different companies can come up with different manufacturing techniques. There's definitely new technologies always happening, different purification methods, different modifications of the cell lines, different manufacturing sites. There's all this variability when producing these. So every time they make a change in their manufacturing process, they kind of have to go through the process to get it reapproved, or to at least to prove to Health, not to get it approved as a new drug, but to get it, they have to show Health Canada that when they do make a change in their production, that the efficacy and the safety of their drug that they're producing is maintained. So if you look at an example here on the graph on the right, so if you look at infliximab, so this was in 2016, this is from a reference in 2016. So at the time, so infliximab had 50 different manufacturing changes to how they made their drug. And every time they did that, and some of them were high-risk changes, as they call it, which would, if you modify how you make it, they still have to show that what you're producing is the same. Because as I've said, it's not always the same drug being produced exactly, because it's this big heterogeneous kind of proteins and glycoproteins and stuff like that. Now, when a reference biologic medicine, so basically when they do make those changes, they're not doing phase 3 clinical trials over and over each time, but they just have to do specific analytical testing or non-clinical studies, so things like PK/PD kind of studies to show that the drug still has the exact same effect since they made these modifications. And it's the bottom line here is that the scientific principles for establishing biosimilarity are the exact same as for demonstrating comparability after a change in the manufacturing process of an already licensed drug. So basically, any company that's coming in and that's making another version of this drug, they're going through the same thing that the originator company has to do if they make a change in their process. And it's just that, according to the rules of Health Canada, the biosimilar must be within 20% of the biologic in various criteria. So these are often PK, PD, immunogenicity, and stuff like that. So they have to be within a range of safety, and that's enough to show that the drug will maintain its efficacy, its safety, and its quality. And so what are the regulatory requirements? So we'll start with my final test question for you guys. So the concept of totality of evidence required when validating a biosimilar compared to a reference drug does not require which of the following aspects. So proof that the biosimilar is manufactured to the same regulatory standards. Biosimilar is established to be highly similar. The biosimilar exhibited no clinically meaningful differences in efficacy and safety in comparison to its reference drug. Or D, phase 3 trial with large numbers showing efficacy and safety. And so, yeah, so 68% of you said it was that phase 3 trials, so basically phase 3 trials are not needed. That is the correct answer here. So the safety and the efficacy of the drug has actually already been established by the originator, by the reference product. So it's already been proven. So it's enough to show that the drug is similar enough that you can then bring it to market and you're expected to have the same effects. So this whole process is regulated by Health Canada. There's the Food and Drugs Act, the Food and Drugs Regulations. So basically, the manufacturer has the onus to provide the totality of the evidence, validating the biosimilar compared to its reference drug, so that it's manufactured by the same regulatory standards, that it's established itself to be highly similar, that it's exhibited no clinically meaningful differences in efficacy or safety in comparison to the reference drug. And once all those steps is done, then Health Canada is able to go and give approval for these drugs. And I'll go over this quickly. It's basically just to show you, if you look at the middle graph here, so this is when an originator drug is being brought to market, the steps are very different. So for them, they start with a bit of analytical stuff and then some preclinical, and then they get into their clinical, phase 1, phase 2, phase 3 trials. On the right, when you're looking at a biosimilar, it's actually the opposite. So the emphasis is really on the analytical and the preclinical steps in production, and that's what they have to sort of spend their money on to be able to show that it's highly similar to the reference drug. So those are the big things. And if you take a look, so it comes down to the PK/PD parameters that are really the most important. So an example, if you're going to, Dr. Carrier shortly is going to talk about enoxaparin. So if you're looking at these PK/PD graphs, so if you're comparing one of these biosimilars to Lovenox with one of the new ones, your anti-10A curves should kind of match up with these curves that you would get. So it's things like that. And once you've done all that, you've basically shown that your drug is highly similar and it's ready to go to market. So in summary, manufacturers must demonstrate the similarity of their product to the Canadian reference product by using comparative studies via stepwise approach. The type of data required to support biosimilar authorization differs from that required for a standalone biologic drug or a new drug submission. And the manufacturers must independently demonstrate the biosimilar against a previously authorized reference product. So demonstrate the quality of the drug, perform comparative studies to demonstrate its similarity in structure, function, efficacy, and safety. And then, of course, PK/PD studies become the most important. And with that, I will pass it on to Dr. Carrier, who will talk about thrombosis and where biosimilars fit in here.
Thank you very much, Ryan. This was a really good and comprehensive review of the whole field of biosimilars and generics. And my goal will be really trying to incorporate these notions into low molecular weight heparin more specifically. And it may be good to start with a patient scenario. So we have Etienne here. He's a 67-year-old male with metastatic lung carcinoma. Unfortunately, his course was complicated by segmental right upper lobe PE. He was initiated on a directal anticoagulant, and then 2 months within his treatment on a directal anticoagulant, presents to the emergency room again with right lower extremity swelling, erythema, and tenderness, new progressive shortness of breath. He has an ultrasound that shows a right lower extremity deep vein thrombosis, which is occlusive, and a repeated CTPA that shows new filling defects in pulmonary arteries that were not previously affected. And therefore, patient has been compliant with a medication. After history physical examination, it's deemed that he had a breakthrough event despite anticoagulation. And the next step is what we would do. And there's no doubt that this is a challenge and certainly an unmet need for which more data would be welcome. But if you look at clinical practice guidelines, some guidance, they would suggest that if you had a recurrent VTE despite anticoagulation, while on an oral anticoagulation, maybe consideration of a parenteral agent may be desirable. And in Canada, we have different low molecular weight heparins. Would we feel comfortable with enoxaparin, tenzaparin, dalteparin at therapeutic dosing? I think so. Then the question that needs reflection is for one of the low molecular weight heparin for enoxaparin, we now have biosimilars that are available. Would you feel comfortable using a biosimilar in that particular context? So I would certainly feel comfortable using enoxaparin BID in that setting and feel comfortable with tenzaparin, dalteparin. We'll try to incorporate what was said by Ryan before, and then we'll open up for more discussion. So I think it's always helpful to have an historical perspective about things. And if you think about the different low molecular weight heparin, we'll focus specifically on enoxaparin because this is the only low molecular weight heparin that has biosimilars in Canada. And when we think about the originator of enoxaparin in Canada and the United States would be Lovenox, elsewhere in the world would be Clexane. Well, if you think about when it was introduced, in 1993, the originator of enoxaparin, Lovenox, is approved in the US through a new drug application. And it took all the way down to 2010 to have the first generic enoxaparin, which was made by Sandoz at the time, approved in the US through an abbreviated new drug application. Now, you're probably just thinking, well, Ryan just said that we would have a biosimilar low molecular weight heparin. Mark, you're talking about a generic enoxaparin. Well, it's because the FDA made a statement that if a generic product contains the same active ingredient, they will consider it as a generic drug, and therefore they consider in the United States only low molecular weight heparin as semi-synthetic drugs. So what does that mean? It means that if they're going for approval, they have to look at physical, chemical characteristics of the enoxaparin, obviously, or the low molecular weight heparin, nature of the heparin material, the chemical process, how it's break down into smaller pieces, the nature of the arrangement, a component, specific lab measurements, and specific aspects, like, for example, heparin-induced thrombocytopenia and other drugs' effects on human. But they don't necessarily need to have clinical data in order to have a generic medication in the United States. Now, the biosimilar process is a little bit different. So when you think about generic, the generic enoxaparin in the United States have the similar disaccharide, the phospho-containing tetracycloride, which are important for the anticoagulation effect, but they're not exactly the same, right? It's really the active component that is similar. There are some dissimilarities in composition between the different generic low molecular weight heparin, mostly on subtleties on antithrombin binding. These differences are much lower or much smaller than the difference between the 8 brands of low molecular weight heparin that we have around the world, and I'll come back to that. But the clinical relevance of these small differences between the different types of enoxaparin may be unclear and possibly not that clinically relevant, so we need to be mindful of. So that's the US way to do things. In Europe and Canada, we wanted to have an additional step, meaning that we have approval, similar approval for low molecular weight heparin, but not through as a generic process, but through a biosimilar process that Ryan took a good chunk of time to provide us with more insight on. So the first biosimilar that was available in Europe was in 2013, and they went through the biosimilar process. So what's the biosimilar process? It's the same thing. So they have a specific aspect of quality comparisons. There's non-clinical studies that we talked about. We looked at totality of evidence, but they also add a little bit of clinical studies. The problem is that the clinical studies are not the type of clinical studies that you and I as clinicians were used to see. When we think about clinical studies, we're talking about phase 3 large randomized control trial. When they're looking about biosimilar clinical studies, they're small two-way double-blind studies, usually with healthy volunteers, and they looked at anticoagulated effect with anti-10A, anti-2A, tissue factor pathway inhibitor, for example. And there's also a requirement about phase 4 post-marketing surveillance that is an important component to it. And then when you look at what happened in Canada, so generic enoxaparin in 2010, a few years later, biosimilar enoxaparin in Europe, and then in 2021, we had the first biosimilar enoxaparin that was approved in Canada. And now, as Ryan mentioned, we have 4 different brands of enoxaparin available in Canada. Now, these are very different, so we can't compare the different low molecular weight heparin. They're extremely different. They're not interchangeable. So if you use enoxaparin for a certain indication, it's certainly not the same thing as using dalteparin, tinzaparin, or nadaparin. There's probably a good reason why you're selecting a certain type of low molecular weight heparin. And the reason why they're so different is they have different depolarization processes. Enoxaparin is alkaline beta elimination of unfractionated heparin. Dalteparin is nitrous oxide digestion of producing aminohydrous group. And then tinzaparin is the enzymatic cleavage of heparinase 1. That leads to molecules that are different in molecular weight. They also have differences in the anti-10A and 2A ratio, different half-life. They may have different effect, for example, on renal clearance and other things. So the low molecular weight heparin are certainly not interchangeable, but within the different preparation of low molecular weight heparin, specifically in this case enoxaparin, when we look at generics, can we use different types of enoxaparin and hope to have the same effect is what we're trying to get some insight and knowledge on. When you look at the different biosimilars for enoxaparin, they have all the same indications. So as I mentioned, the first biosimilar in Canada has been available since 2021. And you have now 4 different types of biosimilars: Elinox, Equinox, Nairobi, and Redesca. And they have the same indications that you would see for enoxaparin. So thromboprophylactic dosing in high-risk patients, for example, post-hip and knee arthroplasty, post-high-risk abdominal surgery, thromboprophylaxis in the setting of medical thromboprophylaxis, so patients requiring hospitalization for medical illness. Same thing for treatment of DVT and PE and acute coronary syndrome. They would have the same type of indications. Now, so if we feel like the different types of biosimilars are similar, can we try to compare them and see if there's any characteristics that are making difference? Well, you can see that most of them have very similar findings. A lot of them have been around worldwide in different countries for 5 to 10 years. None of them have phase 3 clinical comparisons. Some of them have post-marketing safety studies in high-risk VT patients, and we'll come back to that in a minute. And then most of them have some of them have in-person device training, some of them have financial support program, education material. So at the end of the day, when you're comparing different enoxaparin, looking at the originator enoxaparin or the different biosimilars, often at the end of the day, you're looking for additional things that may be important to you and your patient. For example, a financial support program may be desirable in certain provinces or for certain patient groups that may not always have coverage, as these low-molecular weight heparin may be important. So if we go back to the patient scenario, and I'll be happy to entertain any discussion at the end of it, when you're looking at patients that has had recurrent VT despite oral anticoagulation requiring an acute indication for parenteral anticoagulation with one of the low-molecular weight heparin, I would feel very comfortable with enoxaparin 1 milligram per kilogram BID in this setting. Therapeutic dosing of dalteparin, therapeutic dosing of tinzaparin would also be reasonable. Now, obviously, as clinicians, there's always more questions, right? We have questions about the low-molecular weight heparin now that it incorporates into the anticoagulation management and practice overall. And we'll try to go through some of these questions together, and if they have, try to come up with answers, and then that will certainly be controversial, and we'll be able to entertain any questions in the Q&A. So some of the questions that are frequent is, why is not every indication of a reference drug studied for a biosimilar? Why are there so few phase 3 trials? From where are the APIs and the source of heparin, and are the low-molecular weight heparin biosimilars safe for my patient in general? So for the first question, why is not every indication for a reference drug studied for a biosimilar? Well, remember, the indication needs to come from the company. So the biosimilar manufacturer chooses which application to apply for, and there's no expectations from Health Canada to force a manufacturer to apply for a specific indication. And some of the indication can't be necessarily be approved because the indication of the original compound may still be under patent. And a good example for that is, for example, on the generic side of things is rivaroxaban, right? So we recently had access to generic rivaroxaban. We can use it for atrial fibrillation and VTE at the 15, the 20, the 10 milligram tablets. But the 2.5 milligram tablets is not available on the generic side because there's the manufacturer, Bayer, still has a patent on it. And therefore, when you're using a rivaroxaban 2.5 milligram, you're actually using Xarelto. Whereas if you're using other dosing, you may be using a generic medication at that time. So because of these two different components, sometimes the biosimilar or the generic medication may not have the approved indication. And again, Health Canada does not force the manufacturer to apply for all this. The next question I hear all the time is, why are there so few phase 3 trials for low-molecular weight heparin? And, people could argue that we need double-blind randomized non-inferiority trials for each indication at therapeutic and prophylactic dosing for all the different indications across VT and acute coronary syndrome. But really, this is not the point. It's not really realistic. You know, if you've done studies that if you do that, you need 1,000 patients in each arm in order to power the trial sufficiency for the different indication, which leads to high costs, which leads to high expenses in RXND, which leads to the biosimilar being as expensive as the brand or the originator biosimilar or the originator molecule. And that's beyond the cost. The reason why we use biosimilar is to reduce cost. And that's why we're using a rigid Health Canada approval in order to avoid these costs so that we can have the product at a lower price and then maybe invest it or have a sustainable healthcare system. From where are the APIs and the source of heparin derived? Well, all the low-molecular heparin available in Canada are derived from pork intestine, and the processes is from unfractionated heparin. I went through the different processes for depolarization across the different molecules. So the reason why we do that is because it's more predictable pharmacokinetic properties compared to unfractionated heparin. So it can be administered at fixed doses or weight-adjusted doses without the need for dose adjustment and other things. Now, other questions that are coming along often is biosimilar enoxaparin must be produced exactly as described in the monograph of the originator product or molecule. Well, I don't think this is possible because the production data are mostly proprietary, so it's confidential. And really, do you really think that we're using the same type of enoxaparin or the enoxaparin derived in the same way in 2023 compared to 1993? No. There's modern production methods, as Ryan highlighted, and that are more precise than the older methods used for the brand or the original product monograph or the original product. So I don't think this is possible or feasible. And sometimes people are saying, well, batch-to-batch analysis demonstrating no difference between biosimilar follow-up and brand enoxaparin again is unlikely to be positive because the biosimilars are now characterized with different analytical strategies than those used for when the brand was launched in 1993. So that would also not necessarily be possible. Now, probably the most important question is, are low-molecular heparin biosimilars and safe for my patients? And as mentioned, some points to consider is that different manufacturing process results in unique chemical structures. Low-molecular heparin and fractionated heparin, they should not be used interchangeably, as I mentioned, but within the certain molecules, I think there's a good enough evidence that we can use them interchangeably, for example, for the different enoxaparin. And then we have pharmacovigilance programs that are available. Obviously, that depends on reporting from healthcare practitioners, but keep in mind that some of these molecules have been around for 5 to 10 years in other countries, and to date, there's not been any signs of concerns or safety-related issues, at least in these particular countries. Now, as I mentioned, it's not the same type of clinical data that is used for biosimilars compared to an original molecule, but there is a little bit of data. So keep in mind that the biosimilar process will ask for biosimilar studies. So there's 4 studies. There are phase 1 crossover studies of healthy volunteers looking at different dosing. And the general conclusion of these 4 analyses is that the different biosimilars were bioequivalent. There's one observational study here that I'm presenting from Italy because they had the similar concerns with biosimilar enoxaparin became available in Italy. So they did a small observational study of efficacy and safety of biosimilar enoxaparin for thromboprophylaxis in high-risk patients. So it's a small study, small number of patients, 381 patients, 89 medical thromboprophylaxis, 192 surgical thromboprophylaxis. They looked at symptomatic confirmed DVT and PE and major bleeding in this setting. And then when they look at the overall effect, again, small numbers, large confidence interval, hypothesis generating, but you can see here that the risk of VT and major bleeding appeared to be similar between patients receiving biosimilar and originator enoxaparin. And since then, there's more studies, right? There's also recently in 2023 in BMC infectious disease, There's a single-arm phase 2 study assessing intermediate dose biosimilar enoxaparin hospitalized patients with moderate or severe COVID-19. Now, this is not a study that compared different brand of biosimilar or compared biosimilar to the originator, but still 301 patients, 98 patients had intermediate dosing, 203 had prophylactic dosing. And the results goes along with what we've seen with other molecules. Originator, enoxaparin, dalteparin, tinzaparin. The risk of major bleeding appeared to be similar between the two groups, and the length of hospitalization seemed to be lower in patients receiving intermediate dosing, which goes along with what we know for the use of low-microwave heparin in hospitalized medically ill patients with COVID-19. And as I mentioned, pharmacovigilance is part of the biosimilar process. It hasn't been available in Canada for many years, but in Europe, there's been a couple of progresses or publication with pharmacovigilance, and there's been no safety signals to date. So I think overall, low-microwave heparin biosimilars are safe for my patient. Points to consider is what Ryan mentioned. There's a large body of confirmatory evidence of biosimilar medicine in all sorts of different indications. But we need to keep in mind that from an enoxaparin or low-microwave heparin perspective, specifically enoxaparin, generic enoxaparin has been used in the US for the past 25 plus years. It's generic, so it can be automatic substitution. Pharmacovigilance has not showed any safety signal. It's available for 5 to 10 years in Europe. Publication are also reassuring. So I think these, although it's a new product and something to consider, and we need to be mindful of it, we need to have a reflection on a patient-by-patient basis, on an institution-by-institution basis. I think these are overall relatively reassuring. So what do you do now that we have, as I mentioned, we had 4 different types of low-microwave heparin not so long ago. Now we have 8 different types, 5 different enoxaparin, plus 3 different types of, well, technically 4 different types of other molecule, tinzaparin, netaparin, dalteparin. So what do we say to patients? So as I mentioned, the different low-microwave heparin are not interchangeable. You wouldn't go from dalteparin to enoxaparin or from enoxaparin to tinzaparin, for example. There's different dosing, different process. There's a reason why we use certain molecules. But if you're looking at enoxaparin and your patient is going home or has been on the originator enoxaparin in the past, and now is going to his retail pharmacy, is getting a biosimilar because in a certain province where it's only covered, how do we explain biosimilar to patient? This is illustrated here. So an 81-year-old male experienced a recent MI, presents an enoxaparin prescription to the pharmacist. The pharmacist will mention that he's given a biosimilar. The patient may not know what it means, but also may have received the originator enoxaparin in the past. How do we explain biosimilar to patient? And that's is important because the different enoxaparin will have different needles a little bit, and we need to, and they're slightly different in the branding and the name. So we need to have key points to share. And I think the key points have been highlighted by Ryan, but biosimilars are highly similar with the same overall effect and similar side effect profile. They're slightly different, but from an anticoagulation perspective, they seem to be having a very similar profile. And biosimilars are reviewed by Health Canada, and they must demonstrate there's no clinically meaningful difference compared to the originator or the original biological agent. And obviously, it's important to have a discussion with the patient, especially if you know or you're suspecting that they'll be switching. They may have been on the originator for a while. Your province may no longer be covering that, so they'll be switched to a different brand. So having a discussion or preemptive discussion about it, having a good working relationship with your pharmacist at the hospital level, but retail pharmacists as well, to know what type of products are available. I think these are things that are important for success. Now, I mentioned that I wanted to keep things brief so that we can have lots of time for discussion. So in summary, all low-microwave heparins are not the same and cannot be used interchangeably. Enoxaparin, dalteparin, tinzaparin, netaparin. They differ in their manufacturing process. They have different molecular weight, which may lead to differences in clinical practice. And you may want to use certain molecule for certain patients. because they have different 10A and 2A activities. But within a certain molecule, all 4 Health Canada approved biosimilars have the same indication as enoxaparin, work in a very similar way. And personally, I don't see any difference in the different types of enoxaparin. There's 13 plus years of biosimilar clinical use, but for low-molecular weight heparin, many countries have been using biosimilars in Europe for 5 to 10 years without any specific concerns, and it may lead to healthcare costs. So these are things that we component to keep in mind when we make a decision on a case-by-case basis, but also within our institution when we have a discussion with the P&T or the pharmacist about what type of low-molecular weight heparin used in different institutions. So we'll open up for questions. Please use the chat. I think there's already some question, and some of them are, the first one is for you, Ryan. So thank you for being so clear on the principle of biosimilar development for pediatric thrombosis. It is not common that specifically for younger age, the number of patients included in trial is very limited. How does Health Canada handle those challenges when it comes to approval of biosimilars in children? That's Leonardo. He's a pediatric hematologist at Sick Kids in Toronto.
Thanks for the question. So I think I glanced over that part. I may have missed that because I had some info on one of the slides on that. So Health Canada can actually demand certain additional studies are done in special populations. So in terms of pediatrics or pregnancy, I don't know, perhaps even very geriatrics or things like that. So before it's approved, and sort of, so I know, like, for instance, with Lovenox isn't, I don't believe it's officially improved. None of the biosimilars or none of the I don't know about enoxaparin. I don't do peds at all, but I believe they don't have the official indication anyways. But I know they're used. So for instance, I know I can speak for Quebec, for instance, the coverage. So what each province has done. So I can, in Quebec as an example, the automatic coverage for a biosimilar actually has a few exceptions, and so sometimes that involves pediatrics, that involves pregnancy, and things like that. So Health Canada's sort of job is to authorize that, but they can actually mention the indications that are officially recognized. So for instance, we're like enoxaparin. So if you look at all the monographs, I believe it says pediatrics is not studied or something like that. So, and they never went after and got that official indication. So it's the same thing that applies to the biosimilars in that context. I don't know if you have anything to add.
Yeah, thank you, Ryan. I'll give you the next question too, because you're so good. So it's an 80-year-old female. It's a little bit different than the topic we talked about, but maybe if you have some insight because you're doing a lot of stewardship and anticoagulation, have you encountered that before? She's an 80-year-old female. She has lower extremity proximal DVT. She started on apixaban. She has itchiness, and then they've tried all the different DOACs, rivaroxaban, edoxaban, dabigatran. She always has the same reaction. Antihistamine is not helping. Have you ever seen or came across that, and how did you deal with it?
So I've had it for individual DOACs. I've never had it where, so we've had it where we've switched and the patient was fine. So I've never personally seen it where it's with all 4 of them and you had that reaction where we were forced to switch, but I've definitely seen it for individual. I don't know if you've seen that.
In a similar way. So I never really had because usually the compound used other than the active ingredient will be different from one brand to the other. It's a bit unusual that, I think we're thinking, is this to like a reaction to the active component of it? It's a bit unusual to also see it with the dabigatran. I never had the chance to do that. Maybe now that we have different generic for apixaban and rivaroxaban, maybe using a different generic might be something to consider. I know for sure that some of my patients on long-term anticoagulation with warfarin, so for example, may have had some intolerance on some of with warfarin, but it was actually some of the strength in the tablet and the coloring of some of the tablets. So switching that around may be helpful. But if she has a strong indication and all the DOACs don't seem to be working, then maybe parenteral anticoagulation with bridging of warfarin might be something to consider in that particular setting, and she needs to have anticoagulation. And I'm not sure if you know this, Ryan, but fonopirinox it's an indirect 10A inhibitor, small molecule, is available in Canada. Is it a generic or is it a biosimilar when it
That's a,
happens?
yeah, that's a good question because I know there are a few, like, because the original company, I think, was it Pfizer, the original Erixtra? I forgot. Because we have, I think it's labeled as a generic, actually. It's a good, it's an excellent question. I'd like to, because I know we have other companies doing it now, and we have it, in fact, in our hospital, we have another brand. I forgot which company it is, but I'm not sure, actually.
Okay, I'm not sure actually either. We don't use a lot of Flunitrazepam locally. We mostly use a low molecular heparin or IV infusion heparin in the setting of acute coronary syndrome. So I haven't kept up with the literature as much, but we'll look into it for you. Cedric from BC, Ryan, has a question about, I guess, the margins, or how similar do the PK/PD for 2A 10A inhibition needs to be in order to be deemed to be reasonable?
Yeah, so I know for a lot of the criteria, Health Canada has that 20% rule on for some of the criteria. So it has to be between, it'll be like 80 to, it ends up being 125% similar on either side there. I don't know, there are actually, I had seen this in the past. I don't know, but some criteria they may want like a narrower margin. I don't know offhand for these specifically though. But there is, I remember seeing a while ago a document where they actually listed all of the different parameters and sort of what's involved, but I don't know specifically for the levels, the FT10A or the two.
Yeah, I think Cedric's question, maybe we can get back to you, Cedric, because you're right that there are different levels based on the different tests, and 2A, 10A, tissue factor, pathway inhibitor, and there's actually more that are available or required. So for your specific question about the anti-thrombin, sorry, the NT2A activity or the residual NT2A activity, we would have to get back to you. I'm wondering if that could be dose dependent as well. But we'll look into it, Cedric, and we'll get back to you on that. I don't remember all the criteria for all the different testing that they're looking at, but they're readily available, so we can certainly do that. And again, I get your point that the residual, or to make sure there's an ongoing anticoagulation effect, especially when you're doing therapeutic dosing, is important. And it is important from a PK/PD. But how, is the variance that clinically important when there's millions of people that used it around the world? I'm not too sure if there's slight differences under the area of the curve, if that's clinically important or not, but your point is well taken. Okay, so maybe we'll use this one as the last question, Ryan. Can we use low molecular heparin for mechanical valve other than mitral, and are any indication for warfarin other than renal failure or mitral mechanical? So what are the ongoing indications? So there's actually a nice elegant paper that was recently published on all the indication for warfarin. So warfarin is certainly not dead, and we could post that publication in the chat, or we can distribute it. But do you want to talk about the different indication for warfarin, Ryan? It's right up
Yeah,
here.
so and for those also, there was a great talk given at Thrombosis Canada, I believe it was 2 years ago also, which is probably available on YouTube, and all the old talks are available on YouTube. So there's some really good material there too that discuss this as well. So generally with warfarin right now, I mean, we're generally using it. I mean, I still, even on a given day, and when we have a 637 bed hospital here, and on a given day, I probably have still a good 5-6 patients on average on warfarin. And so we'll really see indications. So valvular AFib is probably a common one. So we know we don't use DOACs for valvular AFib. We use it for patients sometimes with an LV thrombus. So our cardiology colleagues will still use that. We have patients, and of course with mitral valves, or sometimes patients who may have failed the DOAC, or who, let's say, and who don't want injections. Not everyone loves getting injections as well, so that's another population for which we'll see it. Trying to think of others.
Lupus anticoagulant,
Oh
is
yeah,
a
for sure.
arterial event, for example. Yeah, so these are excellent example, Ryan. Thank you. And then just for that particular question, we just need to be mindful of using low rate heparin long-term for anticoagulation with mechanical valves. Remember, there's not a lot of data. Sometimes we use that in the setting of pregnancy, for example. We're usually following levels on a regular basis to make sure that they're in a range. Not that it's been linked to clinical outcomes, but I would be mindful of using that long-term on a regular basis for anticoagulation. And then maybe the last one is at the end of value code for 6 months VT can change my patients to biosimilar and what patient expects cost-wise. So what are the differences in cost between, let's say, the originator and the different biosimilars, Ryan?
I know for us it was in Quebec it was a difference of I don't have the exact numbers here, but it was somewhere about a 30% cost savings,
Yeah,
something
that's what
like
we
that.
saw in Ontario as well.
Yeah,
So Abdullah, you would expect about a 30% decrease in price or cost saving for compared to the originator.
thank you.
Thank you.
Thank you, everybody. Thank you, Mark and Ryan, especially on behalf of Thrombosis Canada.